I thought we would finish up the linear matrix inversion part of the presentation talking about IOP inversion. And so you take a quick look at this equation, which we've been talking about, where the reflectance is proportional to the ratio of the backscattering of the absorption plus backscattering, where again I've broken up the eigenvectors are in blue and the eigenvalues are in red, and the eigenvalues are the magnitudes that we'll be estimating by regression. And the eigenvectors are the spectral shapes associated with the absorption and backscattering components. So my first question is this linear? And I mean, it doesn't appear to be because it's a ratio, it looks complicated, but it is linear. And if you just start pulling things off to the side and start multiplying this by this side, bringing the R over here, and then you can start seeing what the knowns and the unknowns are. And what are our knowns? What do we know in this equation? Which terms are we? RW, right? So we know R, right? We think we know F over Q. What else? Yep, all the water terms, right? So let's sort those together. And so now we have on this side all the things that we think we know, and on this side we have the things that we think we don't know, or that we're trying to estimate. And so that's actually a linearized form of the equation, which means that we can do a multiple linear regression, different from a nonlinear optimization. So there's a bunch of advantages to a linear regression. So one of them is it yields an exact solution. There's one solution to the linear equation, right? And it's fast, which would make it good for image processing. It allows, I'll show you an easy way to compute uncertainties. And we can include uncertainties in our measurements, as well as uncertainties in the spectral shapes, which are the things that I think we're concerned about. So for each eigenvector, and that would be the phytoplankton absorption or the particle backscattering, instead of defining a single spectrum or a single shape or a single slope, we can define a range of spectral shapes. So we could say, I happen to have 10 different phytoplankton absorption spectra associated with the 10 species that are pigment groups that I know apply to this region. Or CDOM in these waters, the slope ranges from minus 0.01 to minus 0.02, and I don't know where it is in there, but those are significant, those changes in slope. Or the shape of the backscattering, that exponential might range from -1 to +1. Sometimes you can have a positive slope in backscattering. So you can define all those ranges. And then for each measured reflectance spectrum, you can perform this linear matrix inversion for every possible combination. And just think of this as a series of nested loops. Hold everything constant but one, and then allow it to do all its variations. Then go through the next one. And so you end up with however many eigenvectors you have, factorial. So it ends up giving you tons and tons of solutions. And then you allow your statistics to determine the most likely answer. So this would be the result for the absorption coefficient for particulate and CDOM. So this would include this is the everything but water absorption at 440. And you will have a histogram of results, right? And so what you see is that it looks pretty broad, but you could come up with a median value and say the most likely value is somewhere here around 0.047 per meter in terms of my eigenvalue. But it could be anywhere from 0.04 to 0.05. That's not bad, 0.04 to 0.05. In terms of, if you think about the broad range of absorption that you might have, that's actually pretty narrow in terms of your estimates. And so you could do the same thing for the BBP, then you could separate phytoplankton and non-algal particles. You see that there's actually maybe a bimodal distribution to A phi. It could be 0.01 or 0.025 with some spread. But it allows you to be pretty darn certain that it's somewhere between 0.01 and 0.03, as opposed to just giving you a single value. And so each reflectance spectrum gets inverted hundreds or thousands of times, depending upon how you run it. And so then when you start to do comparisons, you end up having, for multiple reflectance spectra, each dot on this line is a single measured spectrum. And the error bar is the uncertainty in the retrieval of that. So now you can say, well, how good are these models at predicting, at being able to interpret the reflectance? Well, pretty good for AP and BBP and CDOM plus non-algal particles. A little bit less robust for A phi, probably because the A phi varies so much in spectral shape, right? Compared to what I showed you last week, which is each spectrum gets one answer, right, with no error bar. So this is an incredibly powerful tool. And so I'm giving you software for this to be able to do both. What I will tell you is that you never seem to get as good a fit to the measured reflectance spectra. Once you estimate the eigenvalues and you reconstruct a modeled reflectance spectrum, it never looks as good as the nonlinear inversion. So it doesn't look pretty on your graphs when you're trying to publish your paper, and you can do it this way and your curves fit beautifully. So what do we do with that? Well, I like to do both. I like to do a nonlinear inversion, get an estimate of what I think the eigenvalues are, run the linear matrix inversion, get a sense of the uncertainty, and get a sense of the difference between the exact solution and the optimized solution. Because then you've covered all your bases. And even though you get a really nice fit, you might not really be getting the right answer statistically, or it might be sort of one of these ones on the edges here. Okay. All right. So that's just a quick, yeah.
The difference in the fit is because you use a different cost function, or where does it come from?
Well, it comes from the fact that you're fitting a line as opposed to the nonlinear inversion, and particularly the way that we use it hyperspectrally. It's fitting, it measures the gradient in reflectance. So it does dr d lambda, and then what you're actually fitting are the shoulders. So it's less concerned with the magnitudes and more with the shape, which is why it does so well for phytoplankton, because phytoplankton have lots of peaks, and so you're fitting the changes in slope associated with absorption in the reflectance spectra, as opposed to fitting the magnitude of that curve. At least that's what I think, that's why I think it fits that way. So it might get the magnitudes, but it's not getting the shape as well, yeah.
So in the combined approach, you use the nonlinear to determine these ranges, and afterwards you do the linear?
Yes, I do the nonlinear first because it's my favorite. Well, because it allows for so much interpretation of the slope, and it lets me know whether the model that I'm using at all fits the reflectance spectrum that I've measured. And I think that that's a good first step to do is, do the IOPs fit this environment? So, yeah.
Collin?
Yes.
What you have shown just before, is it on the, I think it's on a simulated data set?
That one was simulated.
Yeah, okay.
Yes. That's the simulated data set from Zhangping Li's 2016 IOCCG report, and they simulated a series of reflectance spectra with some ranges of IOPs that they put in to calculate it, but and that's from this paper. But you're running it now on non-simulated data. It's messier because they assumed a lot of relationships in the simulation of this data set. They had some dependencies on chlorophyll and things that maybe are not so realistic, in my opinion.
Making that point because you may be surprised if you try something like this to have something which is much larger scatter
Yes.
actually.
Yes.
If you use real data.
If you use real data.
And that's obviously always
Yes.
better with simulated.
Exactly. And yeah, and you sort of have to take that. Because part of what's in there are your uncertainties in reflectance, and part of are your uncertainties in your basis vectors. So, but it's honest. And I think the problem is, if you when you publish something like this, your reviewers are like, what? We have such huge error bars. If you publish something like this, they say, oh, what a nice fit, right? And so I think as a community, and Emmanuel and I talk about this a lot, as a community, we have to be really honest. We have to show this because people aren't used to seeing it, and they're quite judgmental. But you know what? You can be much more secure in this answer than in this answer. And so I think as a community, we have to sort of band together and be like, I'm going to show you mine if you show me yours, because this is the reality of how well we can do. And so if we promise NASA, we're going to give you phytoplankton functional types from space, but with these error bars, like that has to be good enough. And it's the best that we can do. But still can be an amazing contribution to the field. So, any other questions? So, of course, I have a question for you, which we just answered, the uncertainties. We really care about them. Because, well, you tell me, are there other reasons why we might care about the uncertainties? What if we have a big uncertainty in our retrieved CDOM? What have we allowed to vary in the CDOM eigenvector?
Slope.
The slope. Yeah. So maybe we didn't include a large enough range in slope. Maybe CDOM isn't really an exponential. Mike Twardarski published a paper where he suggested that there are other analytic functions which may fit the absorption spectrum better. So the uncertainty can actually point us to things that we don't understand. And so that's another reason to go to your uncertainty. If your uncertainties in phytoplankton are very large, you might be missing a species, right? The model's doing its best to fit, but you're not giving it the pigments it needs to fit the spectrum. So uncertainties can also point you in a direction for improvement. Okay, so from the last two of the lectures, the take-home messages are that these inversion tools are really powerful for estimating IOPs from ocean color. And if you do comparisons read all the papers, they're all very similar in approach. But what we say, the devil is in the details. And so look very carefully at how the eigenvectors are defined. Remember, these papers are some 20-something years old as you start to look at the history of them. And there's a lot of evolution through time as we got better and better at actually measuring the eigenvectors. Thinking about whether you have hyperspectral versus multispectral. If you have an overconstrained system and you do a nonlinear optimization, you do a lot better than if you have 3 wavelengths and 3 unknowns. So there's different solution methods, the nonlinear, the optimized nonlinear, the linear matrix inversion, and they all have some advantages and disadvantages. And then that important consideration, which is making sure you have independent data to test your model, and I cannot overstate this enough. I know I've had some colleagues that are working on a paper doing this massive comparison of PFT models. And they've got a data set and of all these reflectances, and they're all simulating them. And I said, oh, so what are you using to validate? And they have no validation data. So they're just comparing models, but without knowing which one is right. So if you don't have this, don't even start. I mean, it's kind of like, because you just don't know if your answer is right. And then being able to test the sensitivity and do the uncertainty calculations.
Collin,
Yeah.
what do you think about, let's say I've got this data set with, 1,000 observations like your colleagues are playing with. What do you say about, say, randomly picking 300 of the 1,000, developing your model with the remaining 700, and using those 300 as a validation set, as opposed to using all 1,000 from here and then having a totally independent data set from somewhere else for validation? Do you have a...
I have a very strong opinion about that.
That's why I asked, but...
So if you take a subset of your data to develop a model and then test it on the data that came from the very similar place, it's not actually independent. Measuring reflectance out here on Tuesday and measuring it on Wednesday are not really independent. And so I think my feeling on the inversion approach is that reflectance is theoretically related to the ratio of backscattering over absorption plus backscattering. And it doesn't matter if you're in the Arctic under the ice. It doesn't, well, yeah, you can't be under the ice, in the ice. It doesn't matter if you're in the tropics. It doesn't matter where you are. That physics holds. And so you should be able to develop a model for a region and test it in a new region. And it should be the same. Now, what might be different is how you've defined your eigenvector. So if you're only defining an eigenvector for Seneca-Coccus because where you're working there's only Seneca-Coccus, well then that's a limitation. But if you develop eigenvectors that are representative of what we see in the world and are independent of that, you didn't collect the absorption spectrum here and then run it in your model, then I think then you can really do a true independent test. But I think subsampling your data, and that's always been one of my issues with neural network approaches, is that you're sort of using your data to tune it and then testing it on the data that you just tuned it with. And so I think it should apply to other regions to be truly independent. So save a whole region, or save a different season, or something that would make it a truly independent. Yeah. But that's just my opinion. Okay, so I'll talk about this. We're going to move now to the practical part. Okay, so we're going to do 3 activities today. We're going to gain some intuition for semi-analytic inversion using Excel. Whoa. And I gather that Zhangping also gave you an example of doing it, so it might be the same. I didn't know that he was doing that too. So we might spend less time than I thought on that. And then we have the semi-analytic inversion, which is the MATLAB code, and we'll work with that. And then Jeremy Wardell wrote a paper in 2013 or 2014 where he called together everyone who would come who was working on inversion methods. And they had a big workshop, a series of them, and he and his colleagues devised what he calls the GIOP, or the Generalized Inherent Optical Property Inversion. And it's in a GUI, it's not in a GUI form. I think it's in a GUI form on the web. I've just given you the MATLAB code for it. But it's a way of basically running a variety of different models for phytoplankton and et cetera, and deciding which kind of optimization. Allows you lots of flexibility. And so I'll leave you with that at the end. And so we'll look at that. Okay. So in the Excel file, go ahead and open up the Excel file. In the Excel file, I've included the OC4 chlorophyll algorithm. I've included the eigenvectors from my 95 paper, and 3 examples of reflectance spectra from blue, green, and pretty yellow water. The first worksheet here is the OC4 algorithm, or chlorophyll algorithm. And so basically, I've just given you what the equation is, defined it, and what the coefficients are associated with that equation. If you go to the next page, it's the eigenvectors. And so what you have is wavelength, you have the absorption, here's absorption and backscattering, absorption by water, here's the phytoplankton absorption spectrum that I've shown you about 1,000 times. Here's the CDOM and non-algal particle absorption spectra. This is if it's combined. You're allowed to define the slopes as you see fit. This is the range that I put in there, but you can change it and actually change the slopes here if you want. Here's the backscattering by water down here, small particles and large particles. You can change the power function. I've done 0 and -1, which are these two. And then if you go to the blue water example, you'll see that there's a measured reflectance spectrum. This is from the oceanic gyre off of Oregon in the subpolar gyre of the North Pacific. This would be the derived the derived absorption spectra that you'll model, and these are the backscatterings. So right now it just shows you water. So what you see is that from the measured spectrum, it's calculated the reflectance ratios for the chlorophyll algorithm. And then it calculates which one of these is the maximal ratio, right, which is what's in the algorithm. And then calculates what it thinks the chlorophyll is. So here the chlorophyll is quite low. It's 0.05 milligrams per meter cubed, which you know because there's a very, very blue spectrum. So anything that's in blue is something that you can change. So you can change the F over Q. I put 0.33 because this is an irradiance reflectance. It's EU over ED. And so the proportionality of F over Q is pretty close to 0.33. But you can change here the contributions of phytoplankton, CDUM, NAP, small and large backscattering. And you're just providing an estimate of what you think their magnitude at 400 nanometers is. And then it will multiply these and start showing up over here. And this modeled curve will start to change in shape. And this calculates the correlation coefficient and the RMS error between the measured and the modeled. And so you fool around trying to figure out which is which. And I think you can do the same. Oh, yeah. So you try changing these values. And then you can do the same with the green water and the yellow water. And then we'll get to the MATLAB code. So why don't we take, I don't know, take 15 minutes. Maybe you can try doing it for all of them, but why don't the people in the front start with blue? Why don't the people in the middle? Why don't you guys start with green, or why don't you guys start with yellow and see how close you can get? Because it takes a bit of fooling around to try to figure out, like, oh, I really need more backscattering in the red. How do I do that? And so give that a try. It'll take a few minutes. It's not a perfect fit, right? Because it seems to me that you need a little more absorption rate in here. But as soon as you add any CDUM, you lose a lot of others. So you can see that if you did a different spectrum, and that's something that I had in this before, where you could begin to look at the difference spectrum, just subtracting. You see that there's sort of little peaks here, there's a little deficit here, little addition here, a little deficit. And so you can begin to see what kind of eigenvector, the shape you might want to vary. So again, this is an open ocean reflectance spectrum, but I used sort of a coastal species for phytoplankton. So maybe it wasn't peaked enough, but it would give you that. So if you look over here, I basically had a very tiny absorption for phytoplankton. I had basically no CDUM, but just a little bit of non-algal particle. Now, does that mean that there was no CDUM? No, it doesn't. It just means that a flatter shape gave a better fit for me. And so you have to be very careful in interpreting these two in terms of the shape, and that just no large particles, but some small particles. And so that's how I got a pretty good error. So if we go to the green water, this is about what I got. Yeah. So, and again, that you see, so this is about 440 right here. So this suggests that the model doesn't have enough absorption at 440, which would mean that the shape of the eigenvector probably needs to be a little bit peakier in order to model that. And then maybe it has a little too much absorption here, but you can begin to, again, use that residual to tell you something about a difference in your phytoplankton spectra. And so the RMS error is pretty low on this one. And in this case, I had, you know, some large particles, some small particles. Now I had a bit more CDOM and some phytoplankton absorption. And then the yellow waters, I got something like this. Now, when I look at the residual of this, I see some different pattern from the previous two. And what I see here is this has one slope here, and this one has another slope. And so that suggests to me that there might be something with the CDOM. Maybe it's not steep enough, or maybe it's too steep. I need something in between. Usually adjusting between NAP and CDOM, you can get the slope you want, but maybe the extremes were not extreme enough. Maybe it was a 0.023 slope for CDOM. So, and of course you see the very nice fluorescence peak out here that you could estimate. So, okay, is that fun? Good, you take that home and play. Okay, so now we're going to switch to the MATLAB codes. And I've given you a range of models. I've given you the 1995 inversion of mine. I've given you Zhangping's 96 inversion, which developed into QAA, so it's probably modified to the 2002 paper. And I've given you the Garver-Siegel-Meritorena model, which Sarah Garver originally published in '97. I've also given you, so I've given you those three. So associated with each, there will be some sort of a driver program, which is where you load your data, your reflectance measurements in. It's going to call the invert function, which is the estimation function. And then the cost function is where you're minimizing the difference between the modeled and the measured. I've also given you some data files for the eigenvectors. So there's one called Phyto average abs, which kind of goes without saying. And then I've given you a series of reflectance spectra, which I've broken up into sort of blue spectra, green spectra, whatever. And then there's no output files. You basically can just save it as you go. So these are the files that I've given you in there. These are the data files. I've also included in there a hydrolyte simulation data set that Kurt generated, I don't know how many years ago. Maybe it says 2013. And associated with that reflectance data set, there's also an Excel file where he defines the inputs that went into hydrolyte. And the reason why this one's important is because then you have a validation data set to go back and see whether your inversion retrieved the magnitudes of the eigenvectors that went into hydrolyte to generate the spectrum. So it's a validation of simulated reflectance, but it allows you to do that step. Okay, so how to start? Open up MATLAB for everyone who's got it. Already open? Okay. And I would go ahead and navigate your Windows Explorer over to the reflectance inversion code folder. And then you need to navigate MATLAB over there as well. And then you'll see all of the files that are available. And the program I want you to open up is the reflectance inversion, and that's Resler-Perry 95, and it'll open up to this. Okay. Everybody has their own way that they like to run. I like to run in chunks myself, because then I can see what each chunk of the code is doing. So I would usually take this chunk and run it, and take the next chunk and run it, as long as I make sure I do a loop. But you can do it however you want. But the first thing that you have to do before you run the program is to figure out which data set you want. And so I think it's defaulted to the West Coast data set, which is the more clear waters off of Oregon and then moving onto the coast. You can also do Deba Bay, which is a fjord in Puget Sound, the Gulf of Maine, and this is actually in Puget Sound where you have the big yellow blooms. And then this is the simulation data set from Kurt. And so what we notice is that the F over Qs are different for them because one is irradiance reflectance, one is radiance reflectance, and so it defaults automatically into providing the correct F over Q for the simulation. So either comment one or uncomment one. So here's some things that I want you to try. And you can open up this file. It's under lecture, it's in that folder, and it will give you the things that I want you to try. So the first question that I have for you is, how well does a sort of low resolution, low spectral resolution, 3-component model fit to measured reflectance spectra? And so you've already selected the data set you want to use. And when you run the program, first select the CWIF wavelengths, okay? Then you're going to select the 3-component model, which is just phytoplankton particles and a combined CDOM non-algal particle, and run the model. And your output will give you a couple of things. It will show you how well your reconstructed spectrum matches the measured one. And it will also give you these retrieved IOPs, and you can look at them. Are those reasonable numbers? So the program prints the following things to the command screen. It's going to give you the eigenvalues for each of the eigenvectors. It's going to give you an estimate of the chlorophyll concentration. And then you can save them into an array or in a spreadsheet so you can compare your runs. The model is in green. This is the retrieved phytoplankton absorption, which is the eigenvalue that you retrieved times the eigenvector that you provided it with, right? Because you've just estimated a magnitude, 0.05 or 0.26, and you have to multiply that times the shape of the curve to get the total retrieved absorption. Is everyone clear on that? Okay. This would be the combined CDM, and here's the backscattering spectrum for particles for that particular spectrum. So you can look at the range of the spectra. I think the first figure is for the very blue spectrum. It's hard, my screen is really squinched, so I can't make it so you can see a big spectrum. But this is a very blue spectrum. And as you go further along to, say, figure 8, that's a much greener spectrum. That's as we got close to the coast, so the maximum was here is around 490 as opposed to over here around 440. Okay. And you can see that the values of the reflectance are quite different too, as are the retrieved concentrations. So this is less than 0.01 and this is closer to 0.05. Okay. And then, I can't remember what figure. Oh, these are the measured spectra. Figure 100 is always the measured spectra, so you can see the range of the spectra that went into each of those. Okay. And then if you look at, and again, my screen is not very illustrative for you. This is these are the absorption by phytoplankton at 443, CDM, and BBP. It's giving you the magnitudes of those, which you could then multiply. But this could be what you would compare for running different models, okay? So you can try it with a variety of different data sets. So we can ask that first question. How well does a low spectral resolution 3-component model fit to the measured reflectance spectrum? What do you think? Is the modeled reflectance close to the measured reflectance in those inversions?
Usually.
Yeah.
But just seeing like some wavelengths that we don't see.
But that's all the wavelengths you have. That's the satellite. So does it fit the measurements? Yeah, it fits quite well. So what this says is that a very, very simple model with only 3 components in it, and varying the level, the relative concentrations of those 3 components, can provide an estimate of a reflectance spectrum over a variety, a large range of values of reflectance, from coastal waters often to the oligotrophic gyre. Just 3 components. And further, that those 3 components only have one shape. An average phytoplankton spectrum, a sort of moderate CDOM NAP slope, and 1 backscattering. So that's remarkable that just changing those in a variety of ways can reconstruct that entire range of reflectance. But that's what you just did in the Excel, right? Except you did it hyperspectrally. But what it says is that the variations in the eigenvectors are small relative to the impact of the changing concentration. So what it says is that the concentration of the eigenvectors or eigenvalues is the dominant source of variability in reflectance, not their shape. So that means it's really robust. And so then as you start to want to retrieve more information, then you need to maybe split non-algal particles in CDOM. Maybe you need to add for a large or small backscattering. Maybe you need to add different phytoplankton. But to first order, all you need are those three. And that's pretty crazy. So, well, it just means that nature is really helpful. So now try increasing the spectral resolution. So run the same data set, do the same 3-component model, but just change to hyperspectral. And now see where things change a little bit. And how does increasing the spectral resolution, does it do better or worse? And do you get the same eigenvalues? One would hope you would get the same eigenvalues. Did you all get that? I did 10 nanometer resolution. So. So what do you think? Did it change significantly, our results? Oh, it certainly made it prettier, right? It's nicer to have a 10 nanometer resolution spectrum. But did it change our estimate of the phytoplankton absorption, the concentration? Well, this one didn't have any wavelengths in here, so that's why it's just estimating from here to here. So that's the CWIFs. But look in here. They're exactly the same. Good. One would hope that increasing the spectral resolution wouldn't significantly change your answer, because otherwise what it meant is they picked the wrong wavelengths for CWIFs, right? They picked the ones that were not providing a lot of information about the phytoplankton. So this is pretty encouraging. It changed a bit the backscattering magnitude and maybe a bit the CDOM, probably because it's fitting a lot more shape, particularly out here where you have more values. Okay, so you can try that with some of the other data sets. And let's see, the next question is, how does the number of retrieved eigenvalues impact your model? And does it change if you increase the spectral resolution? So the fourth thing to try is do the same data set, go back to CWIF's wavelengths, but now increase the number of modeled components. Allow it to separate CDOM and NAP, allow it to separate into large particles and small particles. Now, when you get an estimate of large particle and an estimate of small particle, does it mean that I've divided the particles into two groups? Not really. What it means is that, so I might get some estimate of, that would be the BBP for large particles, which doesn't have a spectral dependence. And this might be my, and that's my estimate, and this might be my estimate of the BBP for small particles. And they have about the same magnitude in this particular retrieval. So does it mean that I have equal amounts? No. What it means is that I really need to add these together, right? So, and I didn't give myself enough space here, but what it means is I have something flatter by adding the two, because it needed this much and this much. And so if I add the two, that's actually the modeled BBP. Well, the cool thing there is then you can estimate eta. You can estimate the power slope of the sum. You can estimate that. And you'll find that it's something between -1 and 0. So it's not quite, so it might be minus 0.7. And that would say, oh, I have moderately sized particles. In some of the examples, you might find BBP of large and BBP of small, which would be down here. Negative. Does that mean I have negative small particles? No. When you add these together, what you get is some, if I have to add them, no, I can't do this. So this is minus here, and this is here. I might get something that looks like this, which is a crazy slope because we haven't talked about that at all. But I will tell you that when you have very large phytoplankton, like a HAB bloom, a harmful algal bloom of very large cells that are relatively monodispersed, you can find yourself in a regime where if you do me modeling to look at the spectrum of backscattering, you can get a slope that's positive. So what this model allows you to do by asking how much of this and how much of this you have to add together to get the right slope, you can actually retrieve something more realistic for your environment. Sorry, increasing with wavelength, yes. Thank you. It's always like doing the math in my head. I'm like, what? Yeah, Kurt just pointed out I did my math wrong. It would be increasing wavelength, not decreasing, sorry. You have some positive CDOM and some negative NAP, and it's giving you essentially 0 for the sum. CDM is the sum of those two, right? Or it's even a little bit negative. So is there really negative absorption by non-algal materials? Probably not. There's probably some in there. But relative to the phytoplankton, it's saying that you don't need any in there to discern or to reconstruct the measured spectrum. So I could interpret it this 2 ways. There's probably some detrital matter in the water. But because it has a slope like this, and because the shape of the phytoplankton curve might, if you looked at the blue to red peak ratio, maybe the actual one is lower, but because the eigenvector we put in had a high slope, that encapsulated all of the CDOM that maybe is in there. So there's a source of uncertainty. We put in a phytoplankton that managed to give us a good fit, but maybe this blue to red peak ratio is not quite the actual one for the organisms in the water. And so there's some little slope of CDOM, or non-algal particles, that's sort of in this shape. Same thing for the backscattering spectrum. For this particular case, it says you don't really need any particle backscattering at all. In fact, it's a little bit negative. Does that mean there are no particles? No, I'm sure there are particles. I've never seen water that didn't have any particles. But this, for this particular example, this is figure 7. So this is coastal, well, it's offshore coast. What it says is that the shape of the water backscattering plus all of this absorption is sufficient to explain this curve. So within our uncertainty, particle backscattering is probably pretty low. But this is where having a sense of the uncertainty is really critical, because maybe the uncertainty is like this, and so actually there is some particle backscattering. So just know that you will kind of always get a solution, but you really have to go back and be looking at your spectra, retrieve spectra, to see what's reasonable. Okay? You'll always get an answer. Okay. So then the question is, go ahead and play, and you can look at different regions and see what types of waters do better with which type of spectra. But the last thing is, there's a critical step here missing from this analysis. And do you know what it is? What? No, it's okay. Don't not say something because it might be wrong. Because if you're thinking it, probably everybody else is.
She said atmospheric correction.
So this is huge. These reflectance were measured at the surface, so they don't include the atmosphere. But if you run these models on MODIS data, or CWIFS data, or whatever data, you'll find some interesting things. Sometimes you find a very big non-algal particle component, or a very big, mostly, backscattering component that's much larger than the backscattering that you might have in the water. And what were you going to say? You were going to raise your hand. It's atmosphere, right? If your atmosphere has not been removed and you have a lot of blue scattering in your reflectance spectra because it's measured from satellite and that has not been atmospherically corrected, it shows up as an in-water component in the inversion, which, if you had validation data and you knew that your particle backscattering should be 0.01 and not 0.1, you can use that to atmospherically correct your reflectance spectra. So this type of analysis can also help you in that respect, particularly in the backscattering spectral shape. It allows you to identify pixels that might have some problems, particularly if you start to see them on an image with a spatial distribution that has scales that are more associated with atmospheric scales of variability compared to oceanic scales of variability. So this can be a very useful technique. So that's a very good point. It wasn't the one I was going to make, but that's an important point. So what happened? So we've retrieved all these values. We've done the measured and the modeled reflectance. They look good. The retrieved values look reasonable in most cases, sometimes not. What haven't we done? There's some errors, so we could do some sort of simulations to get some uncertainties. Absolutely. We could run linear matrix inversion. So that'd be one thing we want to do. Validation, absolutely. What I've not given you is the answers. The measured absorption spectra from FilterPad, the measured backscattering spectra from an EcoBB or BB3 or Hydroscat, the CDOM absorption from a spectrophotometer or an AC meter. We didn't have AC meters when I collected this data. So there was no validation. You don't have the answers. And that's the critical step. And so, if you wanted, you could dig around in my paper and find the answer to these spectra. But the point is, using your spectra, don't ever collect a reflectance spectrum without collecting a validation to go with it, because otherwise you're just making stuff up. So that would be the step that we'd want to do. So right now, we're just running models to look at sensitivity of models. We're not actually validating them. But when we read your papers, when Kurt and I read your papers for review, we'll be looking for the validation for that. So, okay, and I think that's, so the next thing, if you want to compare, there's the program inversion comparison, and that's where we run GSM, QAA, and RP. You can compare these models and think about how the three models differently fit, because they're quite different in their basis vectors. And one of the things that I like to do is in weather forecasting, like if a hurricane is coming, they run a lot of models, right? And then they see, 8 of them said the hurricane was going to hit Florida, and one of them said, oh no, it's going off into the Atlantic. So we can use this also. And so the nice thing is if you have your own model, or you can program the variety of other new models that have come along, you can put them all together and do this comparison and start to gain a sense of uncertainty in that way as well, which is really useful. So, so that's code that I have given you. And then this is the phytoplankton functional group inversion. So it's basically the Roesler-Perry model, but instead of 1 phytoplankton, you can put in 4. So you can give that a try. and see if you do a better job fitting by allowing for different phytoplankton. And here's some questions for you. And then, let's see. I've also given you the code for the Roesler-Boss 2003, which estimates beam attenuation, backscattering ratio, spectral slope of C, and backscattering spectrum. And you can run all these on these same data sets. And then, let's see, what we have until 10:30? Okay, so I'll leave it for you to do those, but I want to show you the generalized ocean model. This is the one from Jeremy Wardell. It's really self-explanatory to run, and there's many options, and it's really easy to modify. So what you're going to do is go over to the GIOP folder. And you can't see it here because my screen is all squished. But basically, there is run GIOP. So if you open this up, and you read through, what you'll see is that you change everything as you go. So it's going to give you an example set of wavelengths, RRS spectrum, and it immediately estimates chlorophyll from that spectrum in the same way that we did in the Excel file. And then you're building up how you're going to parameterize your model. And they have defaults. So it's going to use the air-sea transmission, which is spectrally flat. It's going to use seawater absorption and backscattering. It says it defaults to Pope and Fry for absorption and Morel for 74. But if you have something different in there, you would just define your absorption by water with some other model that you have. Just input it right there. It defaults to the solar zenith angle is 30 degrees. And so you can change these if you want to make it 20 degrees or 40 degrees. You would just uncomment it and type it in. This says that your model and your measured spectrum have to be within 30% of each other. Otherwise, it fails. 30. I don't know why he got 33. But you can use Morel's F over Q, Gordon's F over Q, or you could define your own F over Q. It defaults to Gordon. You can define eta, the power function on backscattering. You can use Lee's or Martorell's, or you can do a user-defined. If you want to put the 2 in, you could do that. Spectral slope of the non-algal particle and Gelb stuff, or CDOM. So you can define your own slope here. This is the part that I particularly like, is it allows you to use Anick-Bracot's model, which is a chlorophyll-dependent model. So it estimates chlorophyll using the OC4 algorithm. Then it says, I'm going to use that chlorophyll to define the absorption spectral shape based on the global models. You can use GSM, or you could use Chiotti and Bracot's, which has the 2 different shapes for microplankton and picoplankton, nanoplankton and microplankton. You can do the nonlinear inversion or the linear matrix inversion. And then it tells you what you should get to the screen just to make sure that you've run it correctly for that data set. So you can also take the data set that I've provided you for all the different waters and get it at whatever wavelength resolution you want and put that in, right here. So you can have it read in those data sets and model the ones that we've just done so that you could do linear matrix inversion on it or a Bracot model on it. So you have everything you need to do that will keep you busy for a few years. And so take it all home if you have questions about it. So make sure you give this a run just so that you know that you're getting it to work. I've modified Jeremy's code a little bit so that you get some figures at the end so you can actually see the graphs. But I think you have everything you need. And you can put all of your own data in here. So I know some of you brought data with you. So you can put your own data in here. You can put your own data in all the other inversion models. It's very straightforward, so you should be good.